I'll say this again when we're back on Facebook, but just so you know, the images that you see on the screen represent the 6 different collections, and the one here on the top left is the Cohen tetrapod collection, and Chris is going to show us some specimens from that. But as you can see, there are lots of there's other collections in the museum as well, and we're going to explore some of these in future Beaty at Home sessions. So next week, for example, we're going to explore the fish collection, which you see.
Okay, we are good. So if you want to re-welcome everyone.
Hey, awesome. So welcome everybody. We're doing our Beaty at Home session today. We're doing it behind the scenes with Chris Stinson, who is the curatorial assistant of mammals, reptiles, and amphibians. And we're about to hear from Chris in just a second, but I just have this image up of the different collections in our museum. We have over 2 million specimens, and they fall into these 6 collections. And just from top left onwards, clockwise, we have the Cohen tetrapod collection. We have marine invertebrates, the herbarium with plants and plant-like organisms. We have the Spencer entomological collection, fish collection, and then fossils. And so I'm going to hand it over to Chris now, and he's going to walk us through the museum, literally walk us through the museum.
Well, is it virtually? I guess.
Okay. Okay, take it away.
Thanks a lot, Kashifa.
No
so I'm
worries.
just going to share my screen here. Thinking about it. All right, as Kashifa said, I'm Chris Stinson. It usually runs together, so I try to say Christopher Stinson rather than people thinking my name is Christensen or something like that. So although this is called Beaty at Home, I'm not at home right now. I'm one of the few who gets to come into the Beaty Museum because I have to look after our critical equipment and make sure all the 2.1 million specimens are in good shape and everything's still functioning in the museum. So this is the background beetles and mammals edition of Beaty at Home. What you see here on this front page is some marten skulls that I study for my master's. I'm actually doing a master's at the same time as working. The middle picture is the teaching collection, which is hidden behind the scenes from the public, one of the few sections of the museum that is not there. That's the mammal teaching collection, the rodents actually. And then there's a cute little picture of an American marten from back in Ontario. So those are the skulls that I study, and that's the live animal. So this is out back of the museum. I can actually see it out the window in the lab that I'm in right now. And this is sort of a first-person shooter, fast view, hyperlapse, 10 times speed of what I do 3 times a week at the Beaty Museum. I might do a little bit of commentary, but it goes too fast to really see anything. So this is entering through the back. Oop, skipped. Let's try it again. I get to check bio-waste containers, the waste streams, some barbecues there. I don't have to check those. Then I'm going downstairs through the back of the museum. I get to check autoclaves, which is part of the Biodiversity Research Center. There's a whole bunch of freezers that I have to go check, and just generally making sure that everything's okay. This is our walk-in freezer where we keep specimens. These are the -80s that are zipping past you here, where we keep our tissue collections. And then regular chest freezers you see where we keep specimens, large specimens, mostly mammals and birds, but there are a few other things in there as well. That's the concrete wall for a little while. This is in the back hallway, or back emergency exit. And this is me going into the collections through the back room. Some of you may find this familiar walking down the aisle. We have an intermittent leak in the roof right there that I like to go check every time that I'm in. There's the whale. Up the ramp, and now to check the most important freezer in the whole thing is the ice cream freezer. Back in through the breezeway into the Beaty and back downstairs, through the curatorial wing. You'll see somebody getting in an elevator. One of the custodians, back into the lab, and to look after the beetle colonies, which are these 3 colonies that zip past right here, and back into the lab, which is what I'm looking at right now. So I'm looking after the critical and living organisms and critical equipment and the living organisms of Beaty. Not too many living things other than beetle colonies, and that's a picture of the bog up in the corner there. That's in the education room. And the beetle colonies, as I said, there's 3 of them, and there's a few people that have plants and fish and things in their offices. Although my partner in crime, Nicola Bailey, is looking after his fish, so I don't have to worry about that. And then on the other side, that's the freezer room that we zip through with my little first person view. Those are the -80 freezers on the right hand side, so we keep our tissue collections in there. One of them actually belongs to one of the researchers upstairs, and then on the left we have a fridge and 3 freezers for our keeping our specimens that are in prep or need to be prepared. And then at the back you can see a little white handle there. That's some plant specimens as well. So we have the 3 -80 ultra low temperature freezers, 11 specimen freezers, the walk-in freezer, which we use for pest management. So anything that comes in to the museum, it has to go through there for 2 weeks first to make sure all the insects are killed or any other pests are killed. Bio-waste freezer was the first one upstairs. That's I just checked that. That's where we keep biological waste that gets picked up and incinerated for the whole biodiversity research center. And then one fridge that we thaw things in, and the ice cream freezer, which I may or may not owe one Reese peanut butter cup to the front desk once we reopen. And then I also checked the collections. You saw me zipping through there really quickly, but this is a view of the collections. So those are fish collection in jars. That's the picture that Kashifa showed at the beginning. Also the fish collection. Checking to make sure there aren't any specimens that are degrading, as well as checking for insect pests. So I go through on a monthly basis and check all the different collections that we have. So we have going from left to right, there's the insect collection in their drawers, cow and tetrapod collection, those are eagles in our drawers. The bryophytes, lichens, mushrooms, and things in the boxes. The next one and the far one is the herbarium sheets. Just to give you a view of behind the scenes of all the different types of cabinets that we have. And I have little glue traps, baited glue traps in most or many of the, not all of them, there's 1,200 and some cabinets, but several hundred of them to sort of spot check in collections for insect pests, because even though we're not here, the collections could still be damaged by things like that. Now into the beetles. So these are my collaborators in the cow and tetrapod collection. Dermestes maculatus, the hide beetle, although they don't seem to like hide very much. Up in the corner is some marten skulls, sort of showing you a progression of how the beetles clean. You can see that the little woolly larvae are in the corner trying to crawl out of that bin, and there's one adult and then a single adult in the corner. They've got these little clubbed antennae with little, almost little fronds on them. They look like little fern fronds, and they have shiny silvery setae underneath. It's kind of like hairs, the adults do, and they're all bristly, and they start out when they're from the egg about 1 millimeter long, and the larvae get quite a bit bigger than the adults. And dermestid beetles are pretty amazing. They've been used in museums for years to clean specimens, but we want to keep them locked up. So we have ours in terraria, double contained within a dispensing room as well. So we don't want these getting out into the collections, as I was just talking about, checking the collections for insects. These are one of the things that would wreak havoc in our collections, munching through all of our years and years worth of specimens. So we wouldn't want that. So they're kept quite a far ways away from the museum specimens. And I have to keep feeding them. Otherwise they will just sort of die off. But they are pretty amazing, because you got to think dermestid, not these in particular, but a lot of the dermestid beetles, they have this neat trick where if they don't have enough food, they can reverse molt. So they molt and then eat their own skin in a time of starvation, in case, because you got to figure that out in the wild where these would normally be, corpses can be few and far between. So you got to have a way to survive. And then this is a time lapse of a similar thing of the beetles munching away on a marten skull. They actually made it so the jaw opened on this one, and you can see the hyoid processes get shifted off. The beetles like to move things around, especially when there's so many thousands of them. And now on to the specimens themselves. So this is me posing for all the mesocarnivore specimens. It's a little bit more than this now. This was about a year ago that I've either prepped, had donated, or gotten volunteers to prepare since I started working at the Beaty. I love mesocarnivores. That's what I study, is the martens. And there's everything from red pandas and raccoons to a mongoose being attacked by 2 snakes in that picture. Like I said, I'm also a grad student, so these are the guys that I'm studying. These are marten skulls. I'm studying the difference between the Pacific marten and the American marten in British Columbia, and using museum specimens to do it. We have about 800 of them in the museum, and I'm doing morphology and also genetic work to try to figure out where the 2 species meet in British Columbia. And what's happening between them, whether they're hybridizing, one's taking over. And throughout their range in the US, there's different things that are happening, but in British Columbia where they meet, it's not exactly sure what's going on. The Pacific marten was just recently re-elevated to its own species again, and American marten is found across most of North America from Alaska to Newfoundland. There's one inside out, just to gross you out a little bit. That's our camera setup. We like to take pictures. It's interesting. You can see the different colors of the skin actually correspond to the different color of their fur as well. And there's a mock-up of me doing measurements. So I have to do about 32 measurements on each skull. And I'm also doing 3D scanning. I don't have any of those cool images yet. I'm still working on getting funding for that. But that's how I'm comparing them with linear morphometrics. So that's a list of all the bones in a skull and sort of an artistic rendering of my work. I thought it was cute. And there's females on the top and males on the bottom. And you can see that the ones on the left are American marten, and then the other 2 are Pacific marten. The Pacific marten has a shorter nose, wider zygomatic arches, so their skull is a bit wider, those arches that go beside basically their cheekbones. And it's a subtle difference, but I noticed it right away. And that's about it for the slideshow.
Chris, we've had a question and some comments.
Okay.
So just sharing, I've had a bunch of positive comments. Melanie saying it's so awesome to see museums behind the scenes. Linda is saying it's cool making your own food by shedding. Not quite an autotroph, but getting closer. So great.
Of course.
And then and then Melanie's asking, do the larva or the adult beetle eat/clean the specimens, or both? And how do you keep the population size controlled?
Well, I'll start off with the adult or the larva. So both of them do eat. The larvae are probably a bit more voracious, and they seem to last a bit longer. They start so small and keep molting up, so they clean things quite a bit quicker. And you can actually use the small ones to clean really small things like hummingbirds and things like that. So the larvae are probably a bit more voracious, but the adults do eat it as well. Just not as much, because they're more interested in reproducing at that point. And what was the second part again?
How do you keep the population size controlled?
So population size control is basically just by limiting their food. I haven't got it up to the point where you would need to cull it or anything like that, but some collections, if you keep putting things in there, there have been instances in the old building that they had to fumigate them because they were bubbling up out of the colonies and things like that, because people just kept putting more and more in. But we just sort of tone back on how much we put in, and they just sort of self-regulate that way. So you don't want to keep putting more and more and more in until it explodes out of the colony. But
Right.
if you limit the food, they limit their population quite well.
Okay. So Melanie is saying thank you. And there's one more question for now. Nicole
Okay.
was asking, do you need to supplement the beetles' diet at all, or do they feed 100% on just cleaning the skeletons?
Usually it's 100% on just cleaning the skeletons. Occasionally I have to put like if we're a little bit short on specimens and things, I might raid the biological waste, because we rough out the specimens before we put them in there. So there's always like a big pile of meat in our freezer to be disposed of. So we put occasionally, like if I can't keep up with them or something like that, or I know I'm going to be away, I might put a chunk of something in there to let them feast on that while I'm trying to get something prepared. But something like a deer that's been roughed out will last all 3 colonies for several months, right? So, because you can only, our colonies are just the small aquaria that you saw, right? So they're not, we can't put them, there's some museums that have entire rooms where you just lay stuff out, but ours is a little bit more modest.
And just a question. Oh, that's great. Thank you. A question from Facebook. Can you define, this is from Maggie, can you define mesocarnivore for us?
It's basically every carnivore that is in the middle ecological niches. It doesn't necessarily depend on size, because some people consider the world's smallest carnivore and the least weasel as a mesocarnivore. They sort of don't occupy the top and end up being mammals, is what it comes down to. I don't know if there's an actual scientific definition of it, but that's sort of the broad hand wavy way of saying it.
Okay, I have 2 more questions from the Zoom chat. I know we're going to look at specimens, but maybe you can do this
Okay.
before. So Linda's asking, how many marten skulls do you have in your collection? And how many skulls do you have to measure for your studies?
Well, I'd like to measure them all, plus some from other museums, depending on where the contact zone ends up being. So currently the Beaty accession specimens, we have 564 marten specimens. Not all of them are skulls, that's just the total. And I have prepared about 1,100 skulls, and there are 250 left in the freezer to be cleaned and sampled and put in the domestic colonies. So what's that? Just under 2,000, I guess, skulls. Okay. A bunch of them are no data specimens that were in our freezer from the Northwest Territories when I first started working at the Beaty. So those won't probably be any use for my study, but they are in the collection nonetheless.
There are 2 more questions
Okay.
in the chat right now. So firstly, Linda says, wow. And then Sheila is saying, I've never noticed this before. Do all carnivores have the lower incisors in front of the upper incisors? And do both, and the other part of her question is, and do both species have similar habitats, or are there differences?
So to answer about the teeth question, so most of them have them like that. It's sort of the way that marten skulls are. I can actually grab one.
This is great. You have one. Yeah.
Oh, that one's the one that has a loose jaw. I'll get the other one. I don't know if you can see it in there. But usually the upper incisors, can you see that? Are sort of overlapping of the lower ones. Martens are kind of interesting. Their lower incisors are 3 sort of put forward and 2 behind them. So it's like kind of buttressed. It's like a zigzag of teeth to help the teeth maintain strength. So it's like their teeth are all crowded, but that's how they naturally occur, and that's how occlusion occurs as well. I don't know, it's pretty small, you can't really see, but does that answer the question? Sort of.
I think so, yeah. The
Yeah,
other part of her question was, do the 2 species
that's right.
of martens have?
So the Pacific marten tends to be along the coast in general. So they, and it's thought that they're, part of the reason that they're separate is that they feed on different things. So they're a bit more generalist and unwilling to stick their heads in holes. Believe it or not, that's a scientific finding. They don't like going into traps, hair snagger traps, or any traps as much as American marten. And it probably has to do with their shorter nose and their narrower or their wider head. So they're more apt to go after bigger things, maybe scavenge a bit more, eat bigger things. So they have bigger jaws as well, and they tend to be larger. But they both like mature forests. It's just probably their diet selection is a bit different rather than everything. And the American marten and Pacific marten do share the same habitat, which is the area that I'm looking at. So it's basically forests everywhere.
There's one more question for now.
Yep.
Melanie's asking, how does the museum select which new specimens to accept? Is there a limit or unwanted species?
Yeah, so there is a limit. It's called how many cabinets we have. But that's the upper limit, although they're talking about expansion, so we might get a bit more. But in the mammal collection particularly, we have about 10 to 15% expansion, so we have to be selective about it. Like I don't want any more raccoons, for instance. They get into wildlife rescue and get hit by cars all the time, and we have a lot of them. And they're quite large too, right? So they take up probably half of a drawer in one of our cabinets. So, and whereas I could fit like 100 mice in there if it was. So we tend to go for things that are more rare. Something that has a lot of data is what limits it. And then eventually it's going to be space when we just can't accept something because there just isn't enough room anymore. We can't fit another blue whale, for instance.
Good point. Awesome. So Sheila and Melanie are both saying, thank you for the answers. Sheila's saying, I think I understand the crowded teeth thing, but we'll have to take a closer look at a skull the next time I can. Nancy, I just wanted to check if there were any Facebook questions.
Not right now, no.
Okay, awesome.
Yeah, thanks.
Okay.
So now it's show and tell time, I guess.
Mhm.
So the specimens that are behind me, I've actually brought out for a loan that I'm doing today, some of them just to show you some neat specimens. I'm sort of attached to my computer, so I'll reach what I can and try not to pull it off the desk. But just some interesting carnivores, mostly carnivores, because that's what I like to study. This guy, anybody know what that is? You can't answer me, but this is a sea otter. I'm drawing a blank. This one's a young one, and he actually has, I don't know if you can see it, but he's got, his milk teeth are still partially in there. So he was young when he died. They're kind of odd in mustelid form. They got quite a blunt nose, and they have these amazing grinding teeth. So that's for grinding up shellfish and things like that. Urchins, things like that. How about I take the big one down and see if I can do it without dropping it.
I'll just say that if people want to chat, guess in the chat, we can do that, and I can pass it on, your guesses.
All right. Yeah, sure. This guy's the biggest carnivoran in the world, the member of order Carnivora. Not the biggest carnivore in the world, because that's something that eats meat, whereas a carnivoran is somebody who's in the order Carnivora. He's got a big nose too. That's a clue. This one is the southern species of it. Any guesses yet, Nancy?
Well, I know it, but nobody else is listening. Go ahead, just go ahead and tell them.
You can tell them. This is a southern elephant seal. It's a male. It's not a super huge male, but they are quite massive. Those are the big front canines that they use to fight other males. And we have one, a species of this, the northern elephant seal, which almost went extinct back in the early 20th century, late 19th century. But these guys are down in like South Georgia Island, Antarctica, and they can get to be I think it's like 21 feet long, whatever that is in meters, and weigh a couple tons at least.
I just gotta say that Maggie did get that, and I didn't see the reply, so she got elephant seal.
Sweet. Good job, Maggie. We also have some skins here. So these are study skins for those of you who haven't seen behind the scenes in the museum. Most of the mammal and bird specimens are like this. So they look like a furry flat sausage. That's so you can get as much scientific information out of them as well as have them easily store. Isn't he cute? None of them have eyes, the study skins, for the most part, because there's cotton in them and it would be prohibitively expensive to put eyes in every single one of our 40,000 mammal or 40,000 tetrapod specimens. And it really doesn't add anything scientifically. Yes, you can get them for like $2 a pair of eyes, but it doesn't, when you add that up, it doesn't really work and it doesn't do anything. So they're all filled with cotton. We also keep their feet bones in to maintain the structure of the feet. And as I'm going forward prepping new specimens, I take the feet bones off of one side so that we have a more complete skeleton. In the past, it was all 4 feet got to stay, but now only 2 do.
There's a question about the elephant seal from Usley. She's asking, how heavy is it?
The skull itself or the animal?
Says how heavy is that large skull?
It's actually fairly light. It's probably about a couple kilograms, maybe 3 kilograms. There's lots of empty space in skulls, right? And like, I'm not saying it's like air light or anything like that, but it's not too bad. I could weigh it. There's a scale over there. Did you anybody want to see anything in particular? I know that people like red pandas. So this is a red panda from the zoo. It was originally at the Stanley Park Zoo before the aquarium became the aquarium that was a zoo. And he's got a kind of a cute face. The red color on the back. The bottoms of his feet are all covered with fur for walking in the snow. And it's just been recently published within the last few months that there's now a second species of red panda. So they've split them apart. I'm not sure which ones we have, and because they were from the zoo, it might not be particularly reliable. It was done genetically, so I'm not sure if anybody's done the morphological work to see if there's an actual difference between them. But now there's 2 species, so maybe we have 2. We have 3 of these in the collection, one of which was wrapped up in a ball with a monkey when I first started working at the museum in the freezer.
Wow. I was going to ask about the feet, because you mentioned that now when you prepare specimens, you only keep 2 feet?
Yeah.
Or like the bones?
Yeah.
Do you do like one front and one back? Or
one front, one
okay.
back. So it's to get at least one side of all the bones, so you can get all the limb bones, right, for a specimen. So it just gives more scientific robustness, right? So that you, with every specimen, because we might only get 1 red panda, we have more, but so you can have an example of every bone, at least from one side, and then it's just the mirror image of the other side. So that's, a lot of museums do that now, and I'm tending towards that. Unless it's for teaching purposes, like if I was to give specimens to you guys, I tend to leave them both on so it looks more aesthetically pleasing and not a little bit creepy. But
there you go.
but scientifically, it just gives you more information. And we take tissue samples of everything. That's the -80 freezers that I was talking about at the beginning.
Cool. We've had a few questions. Nicole said that she was just about to ask for the red panda, and that's what you grabbed. So great coincidence. And then Ashley's asking a follow-up question to her question about the weight of the skull. Yeah. She's asking, would we expect more empty spaces in the skulls of marine mammals or marine animals?
That's a hard question to answer, actually. Um, Definitely the bone tends to be less compact, so you have less compact bone because they don't have to worry about supporting the weight of them. But at the same time, you have the fact that they can grow as heavy as they want because they're supported by water. So you have those two things that are sort of happening at once. These guys, it's probably not too much different from a terrestrial mammal, but maybe a little bit, like 5 to 10%, something like that.
So there's a question for Melanie. Do museums exchange and or borrow specimens to or from other museums regularly, frequently?
Yes. And then I'm done. No, yeah, we do that quite a bit. We've done exchanges actually. Ildiko, my boss and colleague, has set up several exchanges with European museums in France and the Netherlands, I think. And we've traded in the past. Like I have a whole bunch of things from the USSR and Israel that we traded. We sent them a fox from here and they sent a fox back, kind of a thing. And we have a few things from various places. So that's permanent trades, but then loans happen all the time. It gets harder and harder to do loans now because there's so many regulations. You have to have all the permits. It's like a 2-inch stack of papers every time I have to send anything to the US, but lots of researchers request things. A lot of destructive sampling things. So like take a little piece of the fur to do genetic work. We've had people from SFU, from all over, and we've been involved in lots of projects where we lend either the full specimens themselves, like I just sent a whole bunch of skulls just before the pandemic shut down to France again, a whole bunch of collared lemmings so he could do 3D analysis of the teeth forms. So that was the most recent one. And then now when we're getting pictures of everything, we can share data online as well. So the answer is yes, and in multiple ways.
Awesome. So there's, so Melanie's saying thank you for the response. So I think this is more of a suggestion than a question from Sheila. Yep. How about skulls to show teeth comparison of carnivore versus herbivore?
Sure, Sheila, we can do that. Just to make it confusing, I'll bring a baboon, which is an omnivore. It looks like it's a carnivore with those front canines and everything, but that's more for defense. But you can see that their molars are quite similar to ours. They eat mostly plant material, but they will also hunt things in groups or kill things like that. This guy also had one of his teeth that went really bad and broke off there. That was a baboon. And let's see, I'll go for, how about a beaver? Can't get any more plant eater than that, I think. Just eating wood all the time. They've got the ever-growing incisors in the front for chewing, and then the big gap before you get to the cheek teeth. You can take them apart and you can see the prismatic nature of those. Those also continually grow, and they have whorls of enamel with denting in between them so that it wears differentially, and you get that continually rough surface for them to chew wood. And the orange color actually comes from iron to strengthen the front of their teeth, so it differentially sharpens as well. There's another weird plant eater. I'll hold it up for a second to see if anybody has any guesses at what it is. It's not from this continent. It's a bizarre skull. So this is a kangaroo. So they're plant eaters, and as you can see, they have those molars that all lock together so they can chew plant material. And they have a sort of a chisel on the bottom with their incisors. It's somewhat similar to how ungulates, I'll show that next, chew things. So they have those front incisors that they use to cut it off, and they use the lower as sort of a chisel to pull it against. They also use their tongue to do that. The difference being with the ungulates, the deer and things like that. Let's see if I can get this. Is that the deer don't have any teeth on the top. So it's a kind of a, they have got this horny part that's up here that you can't see. It's non-bony, it's sort of a cartilage, and they use their tongue and their lips to sort of shear off. His teeth are a little bit broken, but shear off plant material and then chew it up with the molars in the back. And again, there's a big gap where all of the canines would normally be. There are a few up here of the deer that have a little canine there. Reindeer have them, caribou, elk, things like that. Grab a.
Oh, Chris, I was just
Yeah.
going to ask about that gap. Is it, I've heard before, but I don't know if this is right, that it's that extra extension gives it room for the cud kind of to be? Or no?
Yeah. So they can sort of pass the cud around in there, so they have more room to chew it. They also have fairly expansive cheek pouches to help with that, to bring it back up. But then, and it's also so that most of them have huge tongues, and it's to give them room so the tongue can work and like use the bottom incisors as like a shear off thing. Think of a giraffe. They actually have the prehensile tongue, right, that grabs it, and that's sort of an extreme case, but most other ungulates use their tongue to do that sort of thing as well.
Okay, thank you.
Yeah, no problem. So that is an obligate carnivore, pretty much only eats meat. And you can probably read the label if I flipped it around. This is a cougar or mountain lion, and has a very shortened jaw, shortened nose, and then just has these carnassials. That's where the name carnivore comes from, where these sort of trim like scissors to chew off meat from the bone. And then they've got the incisors and the canines at the front to bite in and kill things, smother things, bite through the back of their neck to hunt.
There's a question for Maggie. I'm not totally sure what this is referring to. She asked, do you cut the whole limb off or skin it?
Oh, when I'm taking the limbs off of the study skins. So it's sort of a combination of the two, actually, to get gruesome. Maggie has prepped many things, so I sort of partially skin it down and then cut the skin and pull the whole thing off, so that there's enough flap of a skin that you can sew it back up so it doesn't look like it's a big gaping hole in the side of it. Because if you just cut right next to the base of the limb, you just end up with a big hole with the cotton coming out. So this way I partially skin it down far enough, cut the skin enough so you can fold it, and then sew it up, and then take the foot off and clean the bones. Well, the beetles do most of the cleaning, but.
Cool. I'm very curious about the specimen at the top, next to the skull, the top of your cart.
Okay, that's the one non-mammal that there is. So just give me a sec. It's a marine iguana from the Galapagos. So we actually, Cowan, our founder that we're named after, he went to the Galapagos and did a collecting trip with Macmillan on their yacht at some point. We've also had several researchers that have done stuff at the Galapagos and have salvaged things, but this was actually on a shelf. Cowan had a TV show back in the 50s, and so this was on a shelf in the back corner behind him when he was hosting it. It's been through a bit of rough stuff. It's actually a bit of a celebrity recently on Netflix, the series of unfortunate events. It's on, if you pause the reptile room at the right time, it's blurry on the back of a shelf back there. But
Very cool. That's awesome.
and go ahead.
Oh, I was just going to say there are two comments about the Linda's saying, nice smile. And Melanie's saying, oh, wow, I thought they were bigger.
Yeah, they're not super huge. That's probably a medium-sized one. They do get a little bit bigger.
I think Maggie's doing a follow-up on the answer to her last question. She's saying, so the paw is missing?
Yes. It's pretty much impossible, especially for small mammals, to try to skin a paw. You just end up mangling the skin. For larger things, we keep them on there, like sort of badger size and up. But anything smaller, it's impossible to skin. The skin is so tight around the digits, and you have the claw punching through it and everything. And even on larger things, you end up punching holes in it just if you don't have the skills to do it. But yeah, we end up tossing the paws out and just putting them through the beetle colony. Especially for small mammals, it's pretty much impossible to skin. You can try, but it basically shreds.
Just to give you an idea, it's about 1:45 now, so we'll do a couple more minutes before we
Okay.
wrap up. Nicole has a question. Is the color of the iguana true to life, or has it faded?
It's faded quite substantially. They're a bit more black than that. So like most of the vibrant pigments, they slowly degrade over time, and more quickly degrade if they're fluid preserved specimens. It just leaches out the pigments entirely. So it would have been quite a bit darker than that. And any of the bright colors, because they often have sort of an orangey speckliness to them, any of the bright colors like that are not stable at all in the long term, so they just degrade.
Cool.
And the mouth was painted.
Are there any last specimens you'd like to show us? Or any last things you'd like to share?
Well, this baby beaver is pretty cute.
Oh my goodness.
And his little feet.
Wow.
He doesn't even have the full guard hairs out yet. He's mostly fuzz. Just trying to think if there's anything else up there. How about the red panda skull as the last one?
Awesome.
So they're in a family all their own within the carnivores, and it's somewhat similar to a raccoon. They're thought to be closely related to them, or fairly closely related, but they don't have nearly the big teeth, and they have quite the set of molars for chewing on bamboo, although they do eat other things. And they have interesting complex molars, and they also have these really wide zygomatic arches. And it's just a odd looking skull when compared to other carnivores. Kind of stubby, really thick, robust for such a small one. Yep, there you go.
Awesome. So we've had a bunch of comments about the baby beaver, saying that it's very cute, it's adorable. And then Sheila is asking if you know the estimated age of the baby beaver.
Not sure about that. He's probably within, it actually might say on it. I'm just checking.
Mm-hmm.
No, it doesn't say, but June 26th. So he's probably a month or so old when he died.
Cool. And then Melanie's asking about the skull. So are the big for the jawbones and muscles, and does the bigger the hole mean a more powerful bite?
So you cut out there the bigger what?
Bigger the quotation mark hole, and I wonder if that's the gap between the arches to the skull.
Oh, so from the zygomatic arches on the panda? Yeah, so that's where the chewing muscles go. So and it has more anchoring in there, and also it has it's kind of washed out, but it has a sagittal crest. So the temporal muscles come up from the jaw. So that's because they eat a lot of bamboo and have to chew a lot. So it may not be like the pound for pound strength, but definitely the endurance of chewing lots of woody fibrous material for a long period of time. So that's probably why they have quite wide zygomatic arches, to keep those muscles in there. But I've never been bitten by a red panda, so I'm not sure how strong their teeth, their bites are.
Okay, well, so Melanie's saying thank you for that answer.
No problem.
Yeah, I just want to check just in this last 2 or 3 minutes that we have, if anyone has any last questions, if there are questions on Facebook.
Okay, I'm just gonna add one from Maggie. It's Maggie asks, are they carnivores if they eat bamboo? No. No, sorry. Well, no,
They're more omnivorous, so they do eat bamboo, but they will eat other things as well. Occasionally other animals, but not very often. Mostly it's a vegetarian diet, but they are carnivorans because they are in the order Carnivora.
So do you want to give a few examples of carnivorans for our audience who might not know this term?
So it's everything that's in order Carnivora. So we have bears, all the canids, so the dogs, the wolves, foxes, then the mustelids, so like the marten, so weasels, mink, badgers, things like that. And then also like seals and sea lions. And then all of the cats, and the cat relations like hyenas and mongoose, I don't know, meerkats, things like that.
Okay, thank you, Chris. Sorry, I didn't mean to answer the question.
No, I enjoyed
Just
it.
popped
That
out. I think that's all from the Facebook side, by the way, Kashifa.
Awesome. Thank you so much, Nancy. I think Chris, if it's okay with you, I think we'll wrap up here.
Yeah, no, that sounds
okay?
good to me.
Awesome. So Linda's just left a comment saying, Chris, you are the best. Appreciate all your knowledge and expertise.
Well, I'm glad everyone enjoyed it. It was fun to break up my day of prepping mammals or marten skulls and pull out some interesting specimens to talk about.
Absolutely. Just before we wrap up, I do just want to show everybody a little slide of what's coming up next week. So I'm just going to share my screen and just wrap up there. Okay. So I just wanted to share this. If you've enjoyed our program today and if you'd like to support the museum, here are some links where you can go and donate, or you can purchase a membership, and the membership will be activated when it's safe to go into the museum again. And then our session next week is going to be about the fish collection with Nicolas Bailey, and it'll be the same thing. It'll be Wednesday, May 27th at 1 p.m. And if you'd like to provide any feedback on the sessions, if there's particular things you'd like to see, if you'd like to see Chris again, you can go fill out the survey and leave that feedback. And with that, we're going to wrap up. Thank you so much, Chris, for your time and for sharing all of the wonderful specimens, sharing all your knowledge.
Well, thank you. It was lots of fun. I hope people got something out of it, and I wasn't too muttery or muffled by my beard, which I keep growing.
Awesome. Okay, great. Lots of thank yous in the comments. Thank you, everybody, for joining us. Okay, great. All right, so have we ended our Facebook? Oh no, we're still live on Facebook.
Oh.
Somebody just needs to make me a host. For some reason it's stopped.
Oh. Okay.
So if you make me a host again, I'll be able to stop it.
Okay. I'm not able to just stop it. I can't see. Okay.
My Kashfa just needs to switch me to
I
whoever.
I was just frozen.
Yeah.
Look.
No problem.
Okay. Yeah.
Cool. Thank you. Thanks, everybody.